Hello, my dear students of class 12 science. Welcome once again to biology class. Today we are going to begin with a new chapter, molecular basis of inheritance, chapter number 6. Okay, so in living organisms we will find two types of nucleic acids. One is DNA, also called deoxyribonucleic acids, and the second one is called RNA, ribonucleic acids. So in almost all organisms, DNA it acts as the genetic material. Like in our case also, we have got the DNA as the genetic material. It stores the genetic information. Clear? With exception in few cases like in riboviruses or in retroviruses, RNA is the genetic material. But in almost all the organisms, it's the RNA which plays an important role. Even DNA has to dependent on RNA for the formation of protein. So it's the RNA, alright, because RNA it acts as the messenger for the protein synthesis. It acts as the adapter molecule to bind the amino acids and to take the amino acid to the site of protein synthesis, and in few cases it also acts as like a catalyst that takes part in chemical reactions. Okay, but when we compare the DNA and RNA, we will see that DNA is much more stable and it is much more suitable for the genetic materials. So this comparison between DNA and RNA, we will be doing it in our further classes. Okay, so today we will learn about the search for genetic material. Why do we call DNA genetic materials? Okay, and how did the biologist came to the conclusion that DNA is the genetic material? So that we are going to learn today. So first of all, we learn the experiment conducted by Frederick Griffith, also called Griffith's experiment or transforming principle. This is the first experiment that we are going to learn today to search for the DNA as the genetic material. So in 1928, okay, so Frederick Griffith, he was a bacteriologist who used to study bacteria. So in 1928, he was doing his observation. He was studying the bacteria called Streptococcus pneumoniae, the bacteria that is responsible for the disease called pneumonia. Okay, so he was working on this particular bacteria, and while working on this bacteria, he observed the transformation principles. Clear? So this particular bacteria, Streptococcus pneumoniae, listen very carefully, it causes disease called pneumonia, and it has got two strains. Okay, it has got two strains. One of the strain of this bacteria has got mucus polysaccharide coat. Okay, has got the polysaccharide capsule, we can say, that will protect this cell of this bacteria from the action of WBC, very white blood cells. And in colonies, they used to form a smooth silene colony. That's why they were called S strain. If you see over here, it's mentioned that smooth silene colony. Okay, so they used to have got this smooth colony. So it's mentioned that smooth silene colony. So they used to have got polysaccharide capsule. So the cell of this particular bacteria, they were protected from the action of WBC. Okay, and they used to be the virulent form. They were virulent. So S strain was the virulent form of Streptococcus pneumoniae, and they used to produce the disease called pneumonia. On the other hand, the other strains of the Streptococcus pneumoniae, they don't have got polysaccharide capsules. Okay, so due to the absence of these polysaccharide capsules, they were easily destroyed by the actions of WBC in the body of the host. Okay, and that's why they are called a virulent strain, and they used to appear rough in appearance or rough in colony, and that's why they were called R colony. You can see over here, they were called R colony or rough colony. So Streptococcus pneumoniae is mainly the bacteria that causes pneumonia, has got two strains. One is called the virulent S strain, okay, and the other one is called the avirulent R strain. So we call it virulent S3 strain and avirulent R2 strain. Clear? So you know now what is Streptococcus pneumoniae and what are the two strains of this particular bacteria. So this we have just discussed. Now let's move further. Now Just look at that, RH strains have got mucus polysaccharide coat Okay, it's not quote, not cost, quote, and we can say polysaccharide capsules, and R strain does not have this. Now further down the experiment, let's talk about. So when Griffith injected mice with the S strain, see very carefully, okay, when Griffith injected mice with the S strain virulent form, the mice suffered from pneumonia, okay, and they died. But when Griffith injected a virulent R strain into the body of the mice did not develop the symptoms of disease pneumonia, and they remain healthy. They did not die. So look at the condition over here. Okay, so we can see over here, when Griffith injected R strain, remember my dear, R strain is non-virulent. They don't cause the disease called pneumonia. Okay, so when Griffith injected R strain into the mice did not suffer from pneumonia, so they remain alive. Clear? In the second case, let's say when Griffith injected S strain, remember S strain is virulent form. They contains polysaccharide capsules which will protect them from the actions of WBC. So that's why they are virulent. They will cause the disease pneumonia. So when S strain was injected into the mice showed this symptom of the pneumonia, and they died due to pneumonia. Clear? Now next one. So Griffith, by applying heat, he was able to kill the S strain of bacteria. Okay, so by applying heat, Griffith was able to kill the bacteria. So he applied heat on the S strain of bacteria. Remember, he applied heat on the S strain of bacteria and killed this S strain of bacteria by heating them up to 60 degrees Celsius. Alright, now after killing this S strain of bacteria by applying heat, he injected this strain into the mice. So look over here, when he injected heat killed S strain into the mice did not suffer from pneumonia, and they were alive. They remain healthier. Okay, so 3 cases we have learned so far. First one, when he injected the non-virulent R strain into the body of the mouse remain alive, remain healthier because it did not suffer from pneumonia. Second case, when Griffith injected S strain, which is virulent, into the body of the mice suffered from pneumonia and they died. Third one, Griffith applied heat and killed the S strain bacteria. Okay, now when he injected this heat killed S strain into the body of the mice did not suffer from pneumonia and they remain healthier and alive. Alright, now in the fourth case, what he had done, he injected a mixture of, listen very carefully, he injected a mixture of heat killed S strain and live R strain, because R strain is non-virulent. Even this heat killed is also non-virulent. Okay, so when he injected the mixture of heat killed S strain and live R strain into the body of the mice, what happened to the mice? Because both of them are non-virulent, and when we go back to the previous one, we learned that when he injected heat killed S strain to the mice did not suffer from pneumonia. When he injected R strain into the body of the mouse did not suffer from pneumonia and remain alive. But what happens when he injected the mixture of live R strain and the heat killed H strain into the body of the mice? Did the mice suffer from pneumonia and died, or did they remain healthier and alive? This is the question for you. Just think of it. The answer. So when he injected this particular mixture of heat killed H strain and the R strain, the mice suffered from pneumonia and they died. How? How did the mice suffer from pneumonia when it was injected with the mixture of heat killed H strain and the R strain? Both of them used to be non-virulent, but how did the mice suffer from pneumonia and they died? Okay, the answer comes over here. He said that somehow the live R strain has been transformed into the virulent H strain. One more time, he concluded that the R strain bacteria, okay, somehow they have been transformed into the S strain bacteria, virulent form, by the heat killed H strain. So he said that certain genetic material was there which was passed from the heat-killed S strain to the R strain, which conformed or which transformed the R strain into the virulent H strain. However, the biochemical nature of genetic material was not defined by this experiment. He just said that there is some genetic material that has been transferred from the heat-killed S strain to the R strain, which was responsible for the transformation of non-virulent R strain into virulent S strain. But which genetic material was that which was responsible for the transformation? This experiment could not define. So this is one of the transformation principle which was done by Griffith. So let's look at the diagram. So if you look over here, when living smooth bacteria which is virulent was injected into the body of the mice suffered from pneumonia and they usually died. Then living R strain bacteria which was non-virulent, when it was injected into the body of the mice remained healthier. They did not suffer from pneumonia. And by applying heat, Griffith killed the S strain bacteria. Now it is called heat killed S bacteria. When it was injected into the body of the mouse remained healthy. And in next one, he injected the mixture of heat killed S strain bacteria and live R bacteria, and when he injected this mixture into the body of the mice suffered from pneumonia and died. Okay, so here comes the main conclusion part of the experiment, that somehow the R strain was transformed into the virulent H strain, but which genetic material was there, it could not explain, because when Griffith isolated the bacteria from these dead mice, okay, he found, like, he mainly observed the presence of virulent H strain and the heat-killed H strains. Clear? So living S bacteria from dead mice was obtained. So how is this possible? How the S strain bacteria, which is virulent form, is obtained from the body of the dead mice? Simple conclusion was that R strain was being conformed or was being transformed into the virulent H strain, but this experiment could not prove that which genetic material was responsible for the transformation. He could not say that. So afterwards, the same experiment was being continued to find out the genetic material. So we learn about this experiment done by Avery, McLeod and McCarthy. Okay, what they have done, they mainly taken those biochemical molecules from that dead mice. Okay, so they have taken DNA, they have taken RNA, proteins from that dead mice, and they mainly exerted heat and placed them separately. And what they have done, they have taken 3 test tubes. Let's say they have taken 3 test tubes, and in each test tube they have taken DNA, RNA and protein. So all these things was taken from the body of that mice, which was killed by injecting the mixture of R strain and the heat-killed H strain. Okay, and from and these biomolecules was then taken separately, okay, and was put in 3 test tubes. So in first test tube, let's say we have got RNA, in second we have got protein, and in third we have got DNA. Clear? So all these things was been extracted. All right, mainly to study that what was the genetic material that was mainly responsible for this transformation principle. Clear? Now, what they have done here, we have to take the proteins. Enzymes, we have to take the enzymes, the protein digesting enzyme, the RNA digesting enzyme, and we have to take the DNA digesting enzyme. The protein digesting enzyme is called the protease, the RNA digesting enzyme is called the RNase, the DNA digesting enzyme is called the DNase. And even we can take the lipase if we have got lipid over there. Okay, so what at first they have taken protease, the protein digesting enzyme. So it was taken in the test tube. So that enzyme protease was taken in the test tube where we have got protein. So protease will digest the protein. Clear? So in the test tube where there is protein, they have added the protease. Protease will digest the protein. In the absence of protein, there was transformation. R strain was being changed into virulent H strain. Now they have taken RNase, the enzyme responsible for digesting RNA. When they have when they have put the RNA into the test tube containing RNA, even there was transformation. Okay, there was transformation which was going on. That means RNA was also not the genetic material. Now they have taken the third enzyme, which is called DNase. DNase means the enzyme which will digest DNA. So when we have taken the DNase and put into test tube that contain DNA, so DNase will digest the DNA. Okay, so those particular test tube in which they have added the DNAs, and that DNA fluidized the DNA. So in that test tube there was no transformation going on. It inhibited the process of digestion, stating that it was DNA that was responsible for transforming the avirulent or the non-virulent R strain into the virulent S strain. So this proves that DNA is the genetic material and it was responsible for carrying on the transformation principle. Okay, so they conclude that DNA is the genetic material. So today we have learned about two experiments to find out the search to find out the genetic material, or we can say search for genetic materials. We have learned about Griffith experiment, also called transforming principles, who was working on Streptococcus pneumoniae, the bacteria that causes pneumonia, and we have learned the experiment done by Avery, McLeod and McCarthy experiment. Okay, in the next class we will learn about the experiment done by Hershey and Chase on bacteriophage to show that DNA is the hereditary material. So this was today's class. Thank you very much. Stay safe, okay, stay healthier and happy learning. Thank you very much. See you in next class.